Thank you so much for coming. I wanted to thank Mikael and Kalle and also Lars, who can't be here today, for all of the organization of today's talk. And of course, I need to thank the Communication Society for giving me the honor of being a Communication Society Distinguished Lecturer. So one thing that Mikael did not reveal, which I'll have to say now, is that you are actually my first talk of my first tour. So we'll have to see how it goes. Don't tell anyone else in Sweden if it doesn't go well, because I have a few more that I've committed to over the next few days. So I suspect that a number of you are here out of sheer curiosity, and it wasn't the draw of the importance of my name, because, you know, what does it mean, biological communication channels? And what's interesting, and perhaps, I don't know, prescient of what happens now, is what I think it was maybe 1995 or 96. I was a young assistant professor. The National Science Foundation in the US would have these mini workshops where people in the field would get together and talk about what the future was going to hold. And I distinctly remember being in one of these meetings, and we were trying to figure out what we should be doing next. And I said, well, what about biological communication? I won't mention any names, but two senior colleagues looked at me and said, that's crazy. We don't do that kind of thing. It's never going to happen. And here I am, you know, more than 20 years later, working on these projects. So what's intriguing about this particular area is that the notion of communication in biological systems is actually not a new one. And for my inaugural editorial, I was doing a little bit of reading, and I thought, let's just play the game of looking at Google Scholar and seeing what happens when you search on molecular communication. And these are a couple of the articles that came up. This is Francois Jacob, who received the Nobel Prize in Medicine and Physiology in 1965 with two other colleagues, and this was the topic of what they won for, the genetics of the bacterial cell. Melvin Calvin was a 1961 Nobel laureate in chemistry, and he got this honor for elucidating some of the processes that make photosynthesis work. One of the things that he was very curious about was, what does it take to create life, right? So if you have certain kinds of compounds made out of carbon, nitrogen, oxygen, et cetera, what is needed to lift that from this compound into something that we could consider to be living? Before I tell you what his conclusion was, this paper is really interesting. I highly recommend that you read it. It talks about molecular communication and the notion of DNA as being storage, coding, if you will, for life. But what's also really curious about this paper is that at some point, there's an analogy being made about one of the processes in bacteria. This paper is basically describing how viruses replicate within bacteria in populations. And so he's talking about an interference process. And the analogy he uses is having two doors that are controlled by a single radio transmitter. So radio communications comes out as the explanatory way of describing how this process works, which I thought was, at least for me personally, kind of a nice coincidence. So what does Calvin say? What's needed? And he says that there are two key things that we need in order to take something from non-living to living. And that is the ability to transfer and transform energy. And secondly, the ability to communicate and also transform information. And so even in the 50s and the 60s, people were worrying about and wondering about how biological systems communicate and how they store information. However, if we looked at somebody who, say, closer to my home, information theory and communications, Claude Shannon was actually not very excited about how he saw things going. So after his 1948 seminal paper, it's perhaps not surprising that many other disciplines thought about what information theory could do for them. And so there were a lot of these papers coming out about what should we do, what should we study. So I hope you'll forgive me because I can't read the whole quote, but this is an editorial from the predecessor to the IT Transactions, the Institute of Radio Engineers Information Theory Transactions. And in this editorial, he says the following: information theory has perhaps ballooned to an importance beyond its actual accomplishments. Our fellow scientists in many different fields, attracted by the fanfare and by the new avenues open to scientific analysis, are using these ideas in, then there's an ellipses, because of course he wants to convey his shock, biology, psychology, linguistics, fundamental physics, economics, and the theory of organization. Although this wave of popularity is certainly pleasant and exciting for those of us working in the field, it carries at the same time an element of danger. While we feel that information theory is indeed a valuable tool in providing fundamental insights into the nature of communication problems and will continue to grow in importance, it is certainly no panacea for the communication engineer or a fortiori anyone else. Seldom do more than a few of nature's secrets give way at one time. So he was extremely skeptical about what information theory could do. Peter Elias has also an editorial subsequent to this where he essentially says the same thing. He conjures up a title of a fake paper and says, "Never write this paper." And it talks about the connection between biology and information theory. Now, so this is in the 60s, right? And so there was this great hope, and then people talked about it, there was a lot of activity, and then it died out. And a question we can ask ourselves is, well, why did it die out? And so Toby Berger, at least within the Information Theory Society, is someone who's very well known for trying to make these connections between biological systems and information theory, in particular neural systems. And in his 2002 Shannon lecture, which the title is given here, and it's meant to be like a quadruple entendre. One is, because we debate this often, is information theory dead? So the implication is no, information theory is alive and well and living. But also, what can information theory say about living systems? All right? And the thesis he puts forth in this lecture is the following, and that is, in the 1960s, we were inhibited by our ability to measure processes, systems, microscopes, et cetera, were very well formed. And so in a way, we did not have access to the information, the data that we needed in order to draw these kinds of connections. And now here in, what do we call ourselves, the teens or the aughts, or in this century, we now have access to all of this. And now perhaps we are poised to be able to make strong connections between things like all of the theories that we study, whether it be information, communication, or signal processing. And of course, control, because systems biology has had a lot of people who are now actively using control theory in order to analyze them. So what I'm going to do today is take you sort of through a walk of two very different avenues relating to biological systems that we've engaged in. And I came into this sort of by accident, to be honest. And it comes from having people that you really want to work with. And so they have helped create this collaboration. Because I think it will be easier to tell the tale, I'm going to start with what I call engineered systems, which are going to be based on molecular communication. And the question you want to ask here is, if we could build nanomachines that could be designed to be transmitters and receivers, how would they communicate within each other, especially if they're in vivo? And so the question we have to ask, at least I do, with any problem I attack, is what is the channel? And if I have a description of this channel, how should I design and analyze communication systems given this channel? The next one might seem a little more strange, but it's actually how I got into biological systems. I have several colleagues at USC who are microbial scientists. They study bacteria. There are a couple of strains that we tend to study at USC a lot. These colleagues are scattered between biology, physics, and earth sciences. And one of the things that they've been looking at is interconnections between these bacteria. This is actually an image taken out of Mo El-Nagar's lab. And you can see here, so you see these pill-shaped blobs, but what else do you see between them? What does it look like? Joachim, you're in front, so I get to pick on you. Rods or strings, right? It seems to be that these bacteria are connected to each other. And what they've actually done is created a form of a nanowire through the transport of electrons. And what's interesting here is that, and we'll talk about this as we go through the talk, the notion of electron transfer in living cells is well understood, well, I shouldn't say well understood, is more understood than other processes. What's happened very recently is that they've discovered that organisms like bacteria exchange electrons. And in fact, in certain cases, they form, and you'll see this image, a multi-hop network where they're exchanging electrons. And people are trying to understand why this is happening. And so when I first saw these images and others out of Mo's lab, I said, I know how to analyze a multi-hop network. It's something we've been doing for a while. You can actually find papers that we've written where it says multi-hop in it, and we look at relays and things like that. Is it possible for us to take these kinds of ideas and apply them to these problems? And I'll tell you, one of the reasons why we're interested in this, beyond the fact that it's a new channel, it's a new model, it's interesting, is that if we can figure out how to optimize electron transfer by using communication theoretic ideas, we give our colleagues the potential capability of dramatically reducing the time and resources that they need to do the right experiments. And I know that might seem like a very mundane thing, but actually if we can do this, it's going to have huge implications. Beyond the fact of trying to understand how they interact, even bringing down experiment time for these colleagues is big, which is why I think they're talking to us. Because in the end, we as the engineers benefit far more from interacting with them than they might with us, at least at this moment. Right now they're doing us favors, And I think, in fact, our joint papers, I'm not even sure if Mo lists them on his CV or on his webpage because, they're shooting for Science and Nature, and so the IT transactions or a JSAC special issue doesn't have the same sort of cachet. So this is just an image that I took, after doing a little bit of Googling, but I wanted you to have a feel for what these nanomachines might look like. So this is actually a device, a nanomachine, a nanorobot. It doesn't look like any robot that you've probably seen, but what it essentially is a DNA cage that has chemicals in it, medicine if you will. It's led into the bloodstream. It has sensing capabilities. When it senses the presence of key molecules, it opens up and the other chemicals are released. So the idea is that in the future we can potentially target medications so they go where the cancer is, et cetera. And that, of course, is what we all talk about. We're trying to cure cancer. I actually have colleagues who are trying to cure cancer, but I'm not going to pretend that we're doing that. I have very modest goals. What can the theories that I know say about these new systems, right? Because everybody has a cell phone. It's considered to be sort of a mature technology. And who knows, maybe these two worlds will coincide at some point. So let's talk about the first case here. So molecular diffusion communication. This is one visualization of it, and I've been a very bad academic. All of the grad students here don't do what I just did. I did not give attribution. This is an image from colleagues at Georgia Tech, where they've been working on this molecular communication problem for a while. And so you could say potentially what I'll have is a colony of bacteria 1, and maybe that's my transmitting device. They will release chemicals or molecules into the environment that will be received or bound at receiving bacteria. So what do we do here? So we're going to focus on diffusion-based communication, the idea that out of these devices, population of bacteria, nanocage, whatever, chemicals will be released. So certainly with respect to the channel itself, this is a system that doesn't require any infrastructure, right? I don't need a wire. But of course, I do need to design the infrastructure that is the transmitter and the receiver. One of the things that we're going to see is that diffusion-based communication suffers strongly from intersymbol interference, which means signals from one symbol leak over, literally, into symbols from another. And this is something that we want to be able to characterize and therefore do design for. And because of the fact that we want to focus on something that might possibly be implementable, I want to look at encoders and decoders that are extremely simple. And so with the simplest set of tools, can I actually design something that has some hope of functioning in these environments? Okay? And so the first question we need to ask is, what is the channel? Before we ask that, though, we should ask ourselves, how might we possibly convey information? So one possibility is concentrations. I'm sorry, Barry, I keep forgetting to not use this guy. All right, here we go. Is concentration. So you see here a 1, a 0, and a 1. And the idea, of course, is that if you release a lot of molecules, you'll call that one symbol. If you release a few, you'll call it another. Another option is actually to change molecule type. And this is being indicated by the different colored circles here. And you could argue that this is a form of orthogonal communication if you have receivers that are specialized to only receive one kind of molecule. That, of course, has greater overhead because I need to have now transmission systems that have storage for both different molecules as well as receiver systems. The other thing you can do is in timing. I release a molecule, I know something about its release time, I see how long it takes to get there, and by varying the interval between these transmissions, I can also convey information. And there's a really nice active area of research right now going on in the information theory community, which is studying the analysis of these so-called timing channels. They come up a lot. Now, of course, I told you we have to worry about intersymbol interference. So very simply here, I could have molecules that I meant to send at high concentration leaking over here where I have the light pink that forms the ISI. It's possible that during this symbol interval, I would say this could be a high concentration symbol and therefore make a mistake. and this can also happen in these cases. So I'm freely diffusing these molecules into environment. I'm not controlling them. And in fact, what I'm going to do is let Brownian motion take over. So the notion that I have to worry about ISI is not at all surprising to you here, okay? So what are the assumptions I'm going to make? The transmitter is this fixed point source of molecules. I am going to allow for the possibility of there being flow in the fluid, right? So I'm thinking about things like in the bloodstream, et cetera, that I have. I'm going to focus on 1D motion here, but that's only because I can come up with closed form expressions for some key probabilities. You can certainly numerically compute the probabilities that I need for 2D and 3D. That's not a problem. It's just that they're messy. And I assume that these molecules are not going to run into anything unless they run into the receiver, which is what I hope that they will do. So once I'm at the receiver, the molecule is absorbed. So what is my channel model here? So if I assume that I have this Brownian motion, then I can take the model of a Wiener process. So I have these independent Gaussian position increments, which are shown here. V here is the velocity again. This is my time increment. And this D is a parameter that's essentially a function of my environment, right? What's the size of the molecule I'm using? What's the temperature of the fluid, viscosity, everything that's physical. And it's going to determine essentially what the variance is here. So it turns out that you can model things in the following way. So if my information is X, and I label this as the molecule release time, then my received signal Y here is going to be what I send plus a noise source. And this noise source has a particular distribution, which is labeled the inverse Gaussian channel. And it has these two parameters, mu and lambda, which are a function of delta T, D, and V. So we have not done as much on analyzing this particular received signal model. But there is some really nice work by Andrew Eckford at York, who's actually one of the big proponents and early adopters of the study of molecular communications. And then also some very nice information theoretic work by Stefan Moser and Dongning Guo that they've done jointly together. So what does this N look like, this inverse Gaussian distribution? It has this sort of expression here. The only thing that I want you to get from this expression is that it's complicated, all right? And so doing computations with this, analyzing these kinds of distributions is hard, all right? And this is what it looks like here. What I am going to do with this is create a simpler model. So I think it's very worthwhile to study this particular problem, this notion of the inverse Gaussian channel. But as has been revealed, it is just technically very complicated, and we don't have a way of analyzing it easily. And as Stefan is very excellent at, analyzing these complex systems, there are these very complex bounds that you can compute in terms of computing capacity, et cetera. I can take a simpler approach that's going to give me some hope of coming up with some answers in closed form, but I still feel capture the essence of what we're doing here. So what I'm actually going to do is I'm going to look at the probability that a molecule is absorbed within a certain time slot. I'm going to call that pi k. So if I'm in slot k minus 1 here, this pi k is now my hitting probability. And I can compute that very easily from that additive inverse Gaussian distribution from before. Now, how am I going to use this? So this is actually a model I cannot take any credit for having created. My colleagues at Sharif University have, although we did some more development jointly. And so if I now look at my information sequence as being these xi's are going to be rates of molecules leaving the storage, okay? And I'm going to model this, these molecules leaving as a Poisson random variable with this rate. Okay, so now we're getting into a regime where things are a little more computable. And what's interesting here now is that my received signal is also, the number of molecules is Poisson, but it's a linear combination of the rates that I've been sending, all right, where the combination coefficients are these hitting probabilities, and I also allow for the possibility of noise. And you might ask yourself, well, where does noise come from? Well, if we're doing this properly, we're using things that perhaps naturally already occur in the body, not necessarily introducing foreign molecules, because we don't know how that's going to end up. And so it's quite likely that there are molecules of the common type that are present in the environment already. So for the graduate students here, if you look at this expression, what does it look like? Anybody? Undergraduate linear systems? Convolution, excellent. Give this guy a good grade. Okay, so this is it. So yes, that's exactly it. Our rate is a convolution of our transmitted sequence with these hitting probabilities. And so this is our new channel model, the LTI, linear time invariant Poisson channel, all right? So we can interpret our system as my information sequence going through an LTI channel and then through a memoryless Poisson system. Now that I have this characterization, right, it's in terms of things that we know, and we can start crunching through computations and design, et cetera. So what I want to do now is design a transceiver. I know I have these hitting probabilities, and in principle they go on for quite a while, right? Because I have semi-infinite ISI, but the probabilities are going to decay pretty quickly. The other thing I have to worry about here, and I'll get to this again, is this notion of a positive system. I'm not sending negative molecules, right? I'm either sending something or I'm not sending something. And we're going to consider two solution strategies, which again are very simple but have some very clever analysis that the students at Sharif did, which is we're going to consider either doing something classical, which is introducing memory at the receiver, right? We're used to decision feedback equalizers. Or we can also now potentially introduce memory at the transmitter, which we also do now in pre-coding, but here our pre-coding is going to be a little bit different. So there's been a fair amount of work recently looking at this notion of how do I design transceiver systems for molecular communication systems here, all right? The only point that I would like to emphasize is A, lots of papers out there, but the B is that most of these schemes are much more complex than what I'm going to tell you about. So they're good for establishing fundamental limits, sort of speed limits as to what we can do, but in terms of being implemented, you're not going to implement a maximum likelihood sequence detector in a nanomachine, all right? I just don't foresee that happening. Beyond the fact, like, who knows how much power that would consume, and as you know, hot things in your body is not a good idea. Okay, so if we introduce memory at the receiver, this is again the system that we envision here. And the issue here is that I'm actually going to allow this m-bit memory to store anything I like. It could be the decisions that I've made on previous bits, or it could be some function of them. And the reason why I want to keep it very general is I would like to figure out what is the best that I can do, right? So can I figure out the performance associated with the best m-bit memory at the receive side, and then see how a very simple strategy compares to that. So, as I said, for the moment we'll assume that we have binary rates. The memory can store b bits of anything. I have a very general memory function, which is a function of whatever I had before as well as the new observation. And then I'm going to decode my rate by applying some other function to the memory that I have stored and my current received signal. All right, there are actually two memories now that you need to keep track of, the memory in the receiver as well as the memory in the channel, right? Because that's what's introducing my ISI. And these two values may not be the same, because in principle, the memory in the channel is quite large. So we're actually going to use a approximate decoding scheme, which simply says I'm going to threshold, right? So I count a bunch of molecules. Is it bigger than a threshold? I received a 1. If it's lower than the threshold, I received a 0. And we can approximate the interference like a DFE using previous decisions. And you can actually show that this simple decoding structure, given this memory, is near optimal. All right? And the other key thing here is that this threshold is a fixed threshold. I'm not changing it as a function of time or what I think is going on. Okay, so now we can do the flip side, which is that I'm going to design a memory function at the transmitter, and that's going to control the rates that I release. Okay? And we're also going to use a very simple decoder here. Now, as I said, we have these positive systems. I don't have any carrier waveforms. I'm not doing IQ. So if this was the channel model for my simple channel, right? So this is a memory of 1. If I were to invert this, you can see clearly that I have negative elements here. And the point that I'm trying to make is that I can't apply H inverse in my nano receiver because I can only count things. So the question is, what can we do? So one of the things that we can show, and this is a result, is that the optimal thing in terms of transmission is for the 0 to send nothing, and to send some positive rate for 1. But if I can't do ISI mitigation through linear combinations, what am I going to try? So again, we're going to assume this simple decoder at the receiver, which is going to be this threshold decoder. And as you can see here, this is just a bunch of molecules. I'm counting them. And it varies dramatically at the receiver. All right? So I need to do something else. And the idea here is actually very simple. And that is, I'm going to vary the rate for a unit symbol as a function of what I've already sent, so that at the receiver, I'm effectively receiving a constant number of molecules for a 1 and a lower set of molecules for the 0. right? So I'm essentially water filling on the interference that I know I'm going to create, okay? And this is pretty simple to do. It's relatively simple to analyze, right? So here's for memory 1 here. So there's a finite state machine that describes all of the possible symbols you can transmit. And if you want to achieve a certain value constant at the receive side, you can optimize how you vary the rates at the transmitter, all right? Now what's interesting about this is that if I want to analyze what would be the optimal transmitter function, so we're going to consider the case where I have some positive, some memory greater than 1 in the channel, but I'm going to allow myself to have infinite memory at the transmitter, right? So that I know everything that I'm possibly going to send. And I'm also going to assume that the distribution of my interference is fixed and it doesn't depend on my transmitting function. If I then optimize what my transmitting function should be under these idealized conditions, this is what I get. I get some constant minus the interference value that I have, and then scaled by the hitting probability here. This is exactly what we were proposing to do, that we vary what we send as a function of the interference we're going to experience at the receiver so that I get a near constant value or number of molecules at the receive side. And so, this is great. I can also compute a lower bound on this optimal transmitter. Here I'm assuming that I have memory 1, very simple, again, infinite memory at the transmitter. And now the transmitter is basically an infinitely long Markov chain, right? Because I know everything that's being sent. And under this condition, you can prove that for the optimal transmitting function, when you're transmitting the bit 0, you shouldn't send anything, right? So you're only on when you want to send the non-zero bit. So how do these systems compare to each other? So let me try to deconstruct the lines here. So I have an information rate along the x-axis. Remember, I'm varying concentrations here. I have my probability of error along the y-axis here. I was trying to be clever to come up with a cool acronym. On the second paper we have a cool acronym. On the first paper we don't, so I apologize. So the adaptive transmitter is this attract, and the adaptive receiver is just adaptive receiver. And so this black line corresponds to the case where I just don't deal with the interference at all. This green line says I'm going to assume that I know what the average interference is going to be, but that's a fixed number, so I do a little bit better. These two lines here that are close to each other are the adaptive transmitter with differing amounts of memory. So the slightly lower one is that I assume I have infinite memory at the transmitter. This dirty yellow line is assuming I only have 2 symbols of memory. And you can see with 2 symbols of memory, I get so close to the infinite memory at the transmitter. Now, it is absolutely true that if I do the decision feedback equalizer type of structure, I get better performance. But we are conjecturing that actually the adaptive receiver is going to be a higher complexity than the adaptive transmitter. So this gives you an example of the kinds of receiver designs you can do and the things that you might want to worry about. Now, of course, one of the questions you might ask is, given this definition of the LTI Poisson channel, can we say something about the capacity of this system, from an information theoretic point of view? And so we have a series of results on this. They start out very general, which is to say that we have results on a multi-terminal single hop version of this. So now I have multiple information sequences that are going in. They go into this LTI system and then individually go through a memoryless Poisson before you have these kinds of receivers. Whether this is the right generalization is unclear, but we know that in the special case of one transmitter and one receiver, we have exactly the system that we were studying before. You can describe a block memoryless version of this, and that's important because this block memoryless version gives us a way of bounding the capacity of these systems. So we have a result on capacity regions, but just assume that these are now point to point here. So if you look at one of these general memory limited networks here and the block memoryless one, you can come up with bounds that you've probably seen if you looked at early work by Hurt and Massey on ISI channels. And so it's a very similar kind of case where you can look at essentially you use circular convolution to bound what's happening with linear convolution, all right? And then using sort of the classical kind of techniques that we do, you can also come up with an expression for the point-to-point memoryless MLN capacity, which is given here. And then you can further simplify that to get a bound on the capacity that we want. So the key thing is that these expressions are far more computable than what we started out with. But as you can see, they're not all single letter results. So in the end, because I'm conditioning on this sequence, how many symbols I consider increases the complexity of the computation. So I'm not fully satisfied with these results, but they're giving us a step. What we can also do in this case, and I think this actually invites much further investigation, is that, and this is something Amin Gohari came up with. He's an assistant professor at Sharif and one of our collaborators in this team. And he suggested the use of using the symmetrized Kullback-Leibler divergence as an upper bound on the single one. Because we know if we maximize over distributions over the single one, we do get the capacity expression that we need. So clearly, if I'm adding something to it, I'm getting something that's going to be an upper bound. But it turns out the reason why we care about this, right? So I'm getting this upper bound on the capacity expression that I want. What's cool about this is for these LTI Poisson channels, this is computable, all right? Again, if you have studied the capacity of Poisson channels, you'll see that the story is not complete. There are a series of bounds that have been computed. Most of them are highly non-intuitive and take 10 pages of derivation. And what we'd like to do is get something that gives us some insight as to how we should do design. So this, I think, is something that invites a lot more study. So this symmetrized KL is much easier to compute than straight capacity. And it has often a very intuitive feel to it. So for the Gaussian channel, it's actually just the SNR. And for the binary symmetric channel, it has this result here, where again, the entropy of the BSC comes up. And I just wanted to point out this new paper that Igal Sasson wrote recently at the Information Theory Workshop. But he has a lot of nice work on symmetric divergence measures. And so one of the things that's nice about this other thing is that it's also symmetric. And so what we'd like to study is whether we can use Igal's work to potentially further analyze these bounds that we've computed here. So again, I can do a computation, for this case, and I can modify it for the LTI Poisson case. And if you look at this upper bound, it's simply a covariance of two random variables. Now granted, the log complicates things, but it's still more computable than what I had before. So the only thing here, again, that I want you to take away from this is that we have some expressions that we can compute. We believe they will help us get better insight, but we are far from done in terms of understanding how we should analyze these systems. But now I have a name for it, right? LTI Poisson. I have now a way of getting my hands around how I might solve this problem. So what have we done so far? We've been looking at molecular communication via diffusion. We've focused on this modality because we believe it has some hope of actually being used in a real system, because you can design these storage devices to release molecules and receive them. We have this new channel, so I now invite all of you to worry about the LTI Poisson channel. And we looked at some simple transmitter-receiver methods, but I think it's wide open as to what we could do. Also, being biological systems, there are other operators that are open to us that may not exist in our classical, say, RF systems. So for example, Robert Schober, who used to be at the University of British Columbia in Canada and has moved back to Erlangen in Nuremberg, he has a nice a PhD that's just finishing, and I just read the thesis, so I feel confident I can comment on this. But they look at a situation where you can potentially introduce enzymes which have a way of mitigating ISI. Right? And so you have this other sets of possibilities of exploiting what happens in biological systems in order to design good transceivers. So the question that we ask are, what are the other biological features that we can exploit? Okay? And we looked at this computable characterization of a capacity region for this new definition of memory-limited networks. It generalizes previous ISI results as well as subsumes the channel that we care about. We've looked at upper and lower bounds on capacity for these cases, and we have this new upper bound that we think might be very useful. So to be honest, the regime in which I believe it's useful is in the so-called low SNR, which makes sense for us because we're never going to have high SNR in a biological system. But I think again, there's more to be done there. So something that definitely should arise as a question to you that I'm not going to answer is, should there be nonlinear properties that we should be taking into consideration? Because clearly those tend to show up in biological systems. But so far, we've been able to get away without it, And so until we can't get away anymore, we're going to stick to that. So I'm going to switch gears, but before I do, are there any questions about this half? Yes.
You showed us a very intriguing slide of rod-like connectors.
Yes.
But your model, of course, is assuming a random path.
Yes.
Diffusion model.
Yes.
Can't you get a little help from the biologist to establish these connectors between them?
Absolutely. So there are different ways of setting up molecular communication systems. You can create channels using, let's say, natural systems, and there are definitely people that are looking at it. So in a way, your question is perfect because that picture I haven't talked about yet, the one that you pointed out, but that's what I'm going to do next. So there are a lot of different opportunities. The other thing, too, that I think is extremely important to underscore is that the signaling modality that you pick is going to define what your channel and your functions should be. And I think sometimes people believe that there's one way of thinking about this, but I actually don't think that's true. And I think, for example, if you're looking at calcium ion channels, and I can only say that because there are a couple of researchers who've been looking at that specifically as a communication system, it's probably going to have very different properties than if I picked a different kind of molecule or a different kind of fluid environment, right? So these things are extremely specialized, which means in one way it's exciting because we have a tremendous number of opportunities, but in the other way it's hard to compare schemes, right? So if I design a multi-user detector for RF and you do the same thing, I can compete my receiver with yours, right? Where we have the same assumptions, more or less, and the same kind of environment. Here, if you designed a receiver for a particular protein situation or a particular binding event on with a molecule, that could be completely different if someone has something else. So that, I think, is going to challenge our ability to move forward a little bit because we won't all be talking about the same thing, but as I said, there are a lot of opportunities here. Yes?
So now, as you presented it, and maybe this is coming up in the next part, but it seems like you're approaching the problem as if there were a source separation property of these channels.
Yes.
If you want to get something done within a biological system, it seems like you lose a lot by breaking the information from the communication.
Right. So this is something that needs to be studied, right? So I guess the argument I'll put forward here is that now that I know what the channel looks like, or I have a name for it, I can ask myself, given what might be good properties of sources, how these things two match together. And so in a way, your question is actually quite perfect, because if you go back to that 2002 Shannon lecture by Toby Berger, the whole lecture is about matching of channels with sources. And it was just about that time, or maybe a little bit after that, that Michael Gaspar had worked on his PhD thesis work where this notion of channel and source matching came up, right? And so the canonical example there is that Gaussian sources and Gaussian channels are matched, and this is nice, and things are very simple, and so the optimal thing to do is simply scale your signals in a certain way so that you meet SNR requirements. But what's tricky there is if you look at those papers, the cases where they can do the matching are very few, all right? And so we have to figure out what the right thing to do is. But this is exactly what Toby's paper is about. And what's also interesting here is that I'm not actually sure he ever formally published those results. So this information theory newsletter article is actually the source that people are citing right now in terms of those results. I'm negotiating with him right now on an invited paper for the new journal where he's going to take a different view on things, but yeah. Okay. Let's get back to the exciting picture. So this is an image of E. coli. This is the bug that my colleagues study, which is Schuonella onidensis MR1. Took me about a year to get that down, but I have it memorized. Known as Shui for short. Shui is actually of a lot of interest because, well, for a variety of reasons. I think because they're genetically easy to replicate and modify, et cetera. But it also is a bacteria of interest for potential use in microbial fuel cells. And so that's actually one of the reasons why my colleagues study it. We have a number of people who are trying to design these things. Okay, so you can ask yourself, why would you care about bacteria, right? These are these unicellular organisms. They're actually one of the earliest forms of life. In fact, there's some conjecture that it was the large presence of bacteria that actually changed the environment of Earth to enable more complex forms of life to exist. I don't know if this has been fully proven. They outnumber us by a lot, right? So if you took the biomass of all of the bacteria on Earth, it would be larger by quite a bit than everything else, plants, animals, humans, the whole 9 yards. From a research point of view, people study bacteria for a large number of reasons. So again, it's relevant for bioremediation. In fact, the first time I really heard about metal-breathing bacteria, which apparently exists, they respire with metal, it was from a series of researchers at Caltech who were wondering how they could use bacteria to clean up like toxic waste dumps, right? So you have a Superfund site, you can't put anything there because it's dangerous to live there. Can we actually genetically modify bacteria in such a way that they can go in, eat up the bad stuff, and not damage the environment? It's in plant growth, human and animal digestion. I don't know if it's big here in Sweden, but certainly in the US, the gut biome is something that people talk about a lot. There's a lot of popular press on it. Even this notion that just as you have fingerprints that are unique to you, your collection of bacteria in your stomach are also unique to you, and they often define many things in terms of your behavior, including potentially obesity, right? So it's not your fault that you want to eat the hamburger, it's the bacteria's fault. I'm sure it's a part of the equation. Water cycle, carbon cycle, all of these things. Now, despite being these really simple systems, right, single cell, they're incredibly complicated, right? So here's the genome of the E. coli, and you can see there's a lot of stuff going on here. So even though they seem like the one thing we should understand so well, it actually turns out this is not true. Okay, so because you might be asking yourself the following question, what do bacteria have to do with communications? I want to remind ourselves, at least for the canonical starting point if you're an information theorist, is that we usually start from this notion that I have a transmitter and receiver, and what I care about is conveying information from the transmitter to the receiver. And you know, on one hand, this might be happening, right? The bacteria might be exchanging information in order to achieve some kind of goal, and I'll get to that in a minute. But what I want to study here is something different. So I'm actually not looking at communication between bacteria. I want to use communication and information as proxies for something else, and in particular, the amount of electrons that I can transfer, all right? So I go back to this microbial fuel cell example, and as I said, this is a very real research area. People are expending a lot of effort trying to see how they can do this, because essentially microbial fuel cells give you the opportunity to create energy in a non-toxic way, and potentially in a renewable way, right? And this is, of course, something that we're very concerned about. And so the question is, can we take tools from information and communication theory that will help us with this design process? And so the motivating picture that I want to give to you here is actually one from my colleague Ken Nielsen's group out of one of their older papers, and let me see if I can deconstruct this for you. So we have time going along the x-axis here, and we have the current per cell, and when I talk about cell, I'm talking about per bug, all right? So the bugs are growing, they are replicating, they're dying, they're doing all of these things as a function of time, and you're looking at the amount of current being generated by a single bug. And what's very clear here is that there's a sweet spot, right? The population is growing, growing, growing. It actually continues to grow as we go on, but clearly there's a point where too much is not a good thing, right? So they're not increasing the amount of current in a completely linear way. So if I'm designing this fuel cell, this is where I want to operate, right? But I need to figure out what that point is without running, you know, an infinite number of experiments in terms of changing environment, size of the box, and turning all the knobs, right? So we don't want to be knob turners, we want to be optimizers. And so how does it happen? And so it's clear that there's a form of interaction that is occurring on this plate that is somehow inhibiting the production of electrons. And what is that? And you know, so this paper is actually fairly old. I think it's maybe 10 or 15, well, maybe more than that, 20 years old. The point is that we don't know the answer yet, right? So I, as a comm theorist, can walk in and say, well, maybe we can help you answer this. I don't know. I don't want to have that much, I don't know, arrogance, but we'll hopefully learn something along the way. So electron transport is something fundamental to all of us. It happens in each of our cells. It's essentially a way of creating the energy within a cell. So this electron flow creates a proton gradient. That in turn pumps and enables the synthesis, and I can never pronounce these chemicals, so I apologize, of ATP. And so within single cells, we know this happens. But the new thing that's happening now is the recognition that actually there are cells that are one-to-one, you know, connected in a single line. They literally are a multi-hop network. people are beginning to observe these in the environment, and it is typically in water or sludge, if you will. So there are people that go out, they sample the sludge, they take it back to the lab, and they analyze what's going on inside. And they've been discovering these microbial filaments in many, many environments. It's much more prevalent than anyone thought. So why is this interesting or exciting? Why does anyone care? Well, these filaments are really long, like thousands and thousands of bacteria. And the amount of nutrients at the bottom of the cable is very different from the number of nutrients at the top of the cable. So somehow, these bugs have figured out a way to achieve an equilibrium that gives them happiness, all right? And happiness means that you're not dying, so that's always the goal. Not dying is the goal. And so we have this multi-hop communication network from our electron donor to an electron acceptor. And so as I said, when I first saw this, I said, this is a multi-hop cable. Can we see if we can try to analyze this, all right? And one of the things that is new for us is that we have to take into consideration the health of the bacteria, all right? So it's not just having an energy limit or having a fixed battery. It's making sure that you do things in such a way that you don't kill off the organism. And clearly the organisms do this naturally. So one of the things that we need to figure out is how is the health of the bug connected to its ability to transport electrons. And what we're going to use is capacity as a proxy for electron flow. So I want to maximize capacity not because I want to send as much information as I can, but because I want to figure out what's the operating point of one of these microbial fuel cells. Because in principle, the more electrons that I'm transferring, the more electricity I'm going to create. Okay? There's a little bit of a tenuous connection there, but that's the argument that we're going to go with. Okay, so how do we do this? The first thing that we have to do is come up with a model for a single bug. And this is actually the model here, and I'm going to walk you through it. So I should give you some context. People have been trying to model bacterial interaction and behavior for a very long time, all right? What they typically do is take the equations for these chemical processes, like diffusion, et cetera. They have then a series of very long, ordinary differential equations or partial differential equations. If you have multiple bugs, there's interactions. So you can see that this becomes extremely complicated. So in the end, they put all of these things in some big numerical solver. And then when they say that they're doing theoretical work, what they mean is they're recomputing values by turning the knobs on the parameters on one of these big solvers, okay? And that's not what we want to do. I will agree, as you will probably see, that there's still complexity inherent to what I'm doing, but it's not the same level of complexity as the traditional methods, okay? So we want to now abstract away. We're going to use basically queuing theory in order to establish our models. I believe the queues make sense because essentially my electrons are my quanta, right? And I'm collecting these quanta, these packets, if you will. They're coming in, they're going out, there are flows, there are biological constraints that regulate how these flows are going to happen, and then I can try to analyze the system. The fact that we have variation and other small-scale interactions gives me my stochasticity, fine, but there are things that I can do. So as I said, it's still going to be high complexity, and I'm now looking at this, Michael, because you will remember the following, which is the last time you saw me give a talk, or I think it was the last time, I was talking about the use of sparse approximation methods on MDPs. And so my plan for world domination now is that once we get these models worked out, we'll be able to use our methods that we used on these MDPs for this problem here. So as I said, there's high complexity, but I have a plan for addressing it. And I think actually we finally have all of the interworking parts to make this happen. So I'm hoping the next time someone decides I'm distinguished, I can tell you about it. Okay, but what can I tell you about now? So single bug, shown here, I have 4 queues, all right? And you can see that these queues are interconnected. So the first queue is essentially the electron transfer from the previous cell, okay? So that's to be expected. Then I have a so-called internal electron carrier pool. That's also being fed electrons from the environment, so they're also coming in that way. Then I have another queue that regulates how that proton gradient flow is going to regulate my ability to create ATP, okay? So, and then I have the external membrane that goes to the next guy, and I also have leakage. So what's interesting here about these biological systems, so I can't remember who is a good queuing theorist here, but one of the things that you worry about is stability, right? Do the rates match? Do things come in? This is not what bacteria do. they don't go unstable. In fact, they have regulatory processes that essentially say, whoa, too many electrons coming in, okay? So they don't overeat, but they can starve. And again, this is the kind of thing we're trying to assess, right? What are the biological constraints that we can create, add them to our queuing theoretic model, and then see if we come up with something meaningful. Even though this seems kind of obvious and simple, this was the result of much discussion. I think actually maybe it took us a year to sort of agree with the biophysicists that this was the model that they were comfortable with. And this notion of the leakage in and out, that's one of big controversy within their community. And on some level, there should be another flow that we don't have here. Luckily, the paper's already published, but there were experimental results that came out after we wrote the paper that made it clear that we were missing some elements. And this, in a way, for a theorist is fantastic, right? Because if every 2 years someone tells you that the understanding is wrong, that's a new model to be designed and a new system to be analyzed. So we have a lot of work ahead of us. All right, so we have these systems here. I've already talked about how we have this internal pool, which is the NADH pool, molecules diffusing in. We're being a little redundant here. This, again, is this ATP pool, the picture that we saw before, where the electrons cause this proton gradient through electron transfer, and then we have things leaking out. Okay, so we have now this stochastic queuing model that we can use. And the connections that I said before, the dimensions that we need to trade off of each other, are this so-called energy dimension. So again, if the ATP queue is low, then the cell may die. We actually had many funny conversations about how we should define death, and this is what we came up with. But we also have this information type of dimension, which if I'm pumping a lot of electrons to the system, from a comm point of view, this is a good thing, right? More electrons, more capacity, this is great. But actually, if the ATP level gets too high, the system actually slows down. So there's a function that we have to introduce that allows for what we call clogging, all right? So in a way, this is how we're introducing one of those biological nonlinearities, that the function of that the state of that ATP pool governs properties of the channel. And when we go to a capacity analysis of this system, we explicitly model that we have this state dependence that we need to worry about. So we have, on the one hand, been fortunate and unfortunate. So here's a picture of us being fortunate. We were able to find some data in the literature that had to do with yeast and glucose. And there is a whole parameter estimation algorithm that's built into this, but I haven't shown it to you because it's, expectation, maximization, many equations, not very intuitive. But you can see that our computed data, which are the solid lines, match really well with the experimental data we were able to find in the literature. Okay, so what essentially happens is there's an impulse of glucose that's added into the system, and you can measure the amount of ATP that's being generated. And this is how it evolves as a function of time. Because we're looking at a single bug here, we don't have that the two external cues, because I'm not connected to anybody, all right? So this is great. I have a whole bunch of other curves that show very good matching. What's the bad news here? Well, this is not our data. We actually don't care about yeast and glucose. We want to study what's happening with the Shewanella onidensis MR1. And the challenge there is that you can measure one of these quantities, ATP, but you can't measure the other very easily. And so our colleagues are having to design a new experimental setup that's going to allow them to get the other measurement. But without these two measurements, we can't validate our model. But we feel very good about this, and a lot of the biologists who have seen our data matching were impressed. In fact, when we first showed these results to our biophysics colleagues, they didn't believe us. They thought it wasn't true, that there was no way that a model could be as good in terms of data matching, because that's not what they're used to. So it's one of these situations that I think actually by simplifying things, we're able to get a better fit, because we're not, we have enough data to get good parameter estimates. All right, so what I want to do with this now is I want to see if I can use this to drive the generation of a model that allows me to look at that multi-hop network, which is what I started out wanting to know about, okay? So here I have to agree and argue that perhaps we're oversimplifying things, but it did reveal a model and a description for which we could generate some interesting new results. And so the analysis here is quite interesting. Okay, so if I ask any one of the information theory students in the room, what is the capacity of a multi-hop link? you know that the answer is that it's governed by the capacity of the worst case link, all right? And that's typically what happens. This is not what happens in my biological system, right? I have to take the coupling between the bacteria into consideration, and I also have to take the consideration of the fact that I have this so-called clogging going on, right? That if I feed too many electrons into my system, my ability to process the system is going, the process of those electrons is going to slow down. So I have a channel state that is input dependent, right? If the input is too large, the quality of that link is going to go down, right? And if it goes too far down, the whole thing dies. So the idea is, and this is where we do some cheating, is that we're going to take that cable and we're going to actually club it all together into a single entity, all right? Even so, even with these abstractions, even with this major simplification, many conversations with the physicists to make sure that we were making the right kinds of approximations. And so this was also informed by them. So now my abstraction is, I have my input, which is going to go into an encoder. That's my electron signal. And then I have what comes out, okay, also my electron signal. And so the question is, can I come up with a capacity expression for this? Okay. So now, getting closer to the terms that we're used to dealing with, if I look at my channel here, the channel now is the entire cable. I have all of my Qs with their different number of electrons. And what I'm going to actually do is simply ask myself, what is the number of electrons in the cable? And that value, S of t, is going to be my channel state. And that channel state, of course, ends up being a function of my inputs. So my signal is coupled with the energetic state of the cells. And we go back to this terminology that Niccolò came up with, which is I have this energy dimension, which is ATP is low, the cells may die, but I also have this information dimension that if ATP is too high, I'm going to have clogging and the electron transfer is going to slow down. So in order to compute the capacity of the system, and I have to give you the exact model that we're going to use here, we need to observe that there's a fair amount of work in the literature that covers related problems but not the exact thing. The paper that's the most important to us is this Chen and Berger, and that's Toby Berger paper from 2005, where they look at something similar, where there's full channel state information, which we are also going to have, but there's also feedback. One of the things that they show there is the optimality of coding with memory, or Markovian coding, which again, Markov chain, Markovian coding, not too surprising. But they set up their optimization by using dynamic programs, and essentially you can show that the computation of the capacity is the solution to a dynamic program, right? And what's nice about that is I have my queuing theoretic postdoc who knows all about MDPs and dynamic programs. And so there's a whole series of tools that he can use in order to analyze this kind of system. So abstractions are bounding, right? So I'm now going to introduce this clogging function, A of S, which basically says that, you know, if my input is small, then I'm going to let everything through. If my input is large, I'm going to slow things down, all right? I will eventually show that binary rates are optimal, which again is not too surprising given prior literature. And so, you know, I'm either going to have rate 1 in terms of the electrons that I push through the system or rate 2, okay? So this is now what we have. I'm once again going to use these Poisson models because they make sense for the, you know, transmission of these quanta. And so I now have an input which is going to be Poisson with a rate that is a function of the state of the system, and it's the clogging function that I'm going to apply here. So even though I have these two rates, I still have the potential for clogging to go on because of what's going on in the channel, all right? So this changes things. So what are the variations or the improvements that we have over prior work? Weiner in 1988 looked at static Poisson channels. So we have this finite state Markov channel with dynamism, although Chen and Berger have that as well. In the discrete time, Stefan Moser, during his PhD, looked at the capacity of the Poisson channel. But again, very important work, but I would argue hard to understand and certainly a technically challenging piece of work. Okay? So we have this system. We're going to assume a small slot duration as Weiner does. We have channels with state at the encoder. The encoder only controls the intensity. And I keep telling you this, that the state is controlled by the input process. Okay? So I'm going to try to wrap up because you've all been very good. and try to emphasize the quality of the results or their intuitive value rather than the technical details. So again, similar to Chen and Berger, we can show that we can compute capacity using a dynamic program. Basically, our reward is this mutual information that's a function of the state, as well as a function of our control, which is the inputs that we're putting in. We can actually prove that capacity is achieved by feedback-independent inputs. So while Chen and Berger had feedback, we don't actually need it for our system. We can also show that this stationary distribution here only depends on the mean rate, not the actual rate. This mean rate is, of course, conditioned on state. But this now makes things very simple because we can now try to determine our optimal transmitting function. So this is essentially the distribution of the transmitting function by worrying about the mean only. All right? So my capacity expression now looks like this kind of optimization. I can also show that binary signaling is optimal. So even though I had at the beginning the possibility of transmitting a continuum of rates, I don't need that. And that's actually not that hard to show. It relates to the convexity of the key functions, right? So again, for those of you who are familiar with, Markov decision processes and dynamic programs, in a way these results are intuitively pleasing. So how does this all work? This so-called energy dimension is determined by my mean transmission rate. And my information dimension, which is given by this optimization of this function here, means that I only have to optimize over encoding functions with the same mean rate. All right, so I don't need to worry about every possible rate that I'm going to do. I just need the mean rate, and then I figure out, the value I need to get that mean, which is easy because I'm using binary signaling. So a result that I'll show here, and again, in a way, this shows that we're engineers and not necessarily biologists, but a classic question to ask is, optimizing the system is computationally expensive. How would a myopic scheme that only looks ahead one step, how would that work? And one thing you can show, and in a way this is relevant to the biological system, is that the mean rate for the myopic greedy scheme is larger than the mean rate for the optimal scheme. So one of the things that we can predict, right, because we know our optimal scheme is optimizing between this so-called energy and information dimension, is that the myopic scheme is going to suffer because it's going to be greedy and try to get as much information or electrons through the cable as possible. But by doing that, it's going to induce a shutdown in the cable because it's going to exceed that maximum value, right? So you're going to clog more often than you would have before. And that's exactly what you see. So if you look at the achievable rate for the optimal case versus the myopic case, as a function of parameters of this clogging function, you see that we get a loss. But what's nice now is that we know why. The loss that you're getting is precisely because you're being too greedy and pushing too much stuff through the system. Okay? I'm sure we can all think of more classical engineered systems where myopic schemes actually do not perform that much worse than the optimal. And so we often use them because they're practically implementable. All right, so what have we done? We've looked at this stochastic queuing theoretic model for the inner workings of a single bug and then abstracted that to a cable so that we could exploit information theoretic results and characterize more cleanly this coupling between the energy dimension and the information dimension. Clearly, something we have to worry about is how does the bug figure out what the state is and how does it induce that? So right now, my cable result is by taking all of the units and pretending that they're a single unit, right? That's the new model. What we really want to do is figure out if there's a cleaner way of coupling these individual bugs into an actual cable, right? So that these entities are now distinct. And I don't know if we have to do this. I perhaps did not emphasize this enough, but in what we've done so far, I'm assuming that I have perfect channel state information at the transmitter, and obviously that's not going to be true. So if I then have this multi-cell view, how am I going to get CSI at the relay, right, at each one of the subsequent bugs? And this is actually something that we have done a fair amount of work on, which is looking at joint communication estimation over channels with state. Most of our information theoretic results are for a single hop, but I now think I know what we need to do in order to get the multi-hop. So I've talked about some of these things so far, things that we need to worry about. I've got 1D. Well, I sort of have 1D. I'd like to get a better 1D than I have right now. But then the question is, if I go to 2D or 3D, how are we going to handle this? So we'll see how we do this. So in the last couple of minutes, I want to talk about another project that we've just started with a new colleague in physics who also studies bacteria. He's got a different one, Pseudomonas aerugiosa. I think I've got that one straight. And one of the reasons why he studies this is that he's very interested in this phenomenon that's called quorum sensing. And I don't know, has anyone heard of this before? It's been in the popular literature off and on. So this quorum sensing thing is interesting. And actually, for me personally, it's the first time I started to think about biological systems as communication systems. I had read an article about quorum sensing, and to me, it smacked of cooperative communication. So the idea is that you have a collection of bacteria. They are releasing a certain kind of molecule, but they're also sensing the molecule. And once the level of the sensed molecule exceeds a certain level, the bacteria are able to engage in cooperative behaviors that they couldn't do before. And in fact, they express a gene. They change themselves. I mean, they change the next generation. And so people have been very fascinated by this. And in fact, one of the places where you see this is that there's literally, I'll go over here. There's literally a collection of bacteria that start to luminesce when they achieve a quorum. And this is actually the bacteria that this colleague, Ken Nielsen, we saw his current image before. And in a way, he's the grandfather. I mean, he is a grandfather, so I guess I'll call him the grandfather of quorum sensing, and one of the first to really understand the underlying mechanisms. People worry about this because quorum sensing appears to occur in many, many different environments. And one of the environments that we are interested in is actually in disease control. All right, and this is a very big area right now. I'm sure many of you have heard about the concerns of bacteria the fact that many bacteria are becoming immune or resistant to antibiotics. And of course, antibiotics kill bacteria and a lot of other things as well, including good bacteria. And so the question is, can you actually introduce or change the environment in such a way that you prevent these bacteria from quorum sensing? So they're still there, but the disease does not occur because they have not engaged in this new gene expression. And this is a very, very hot topic. And so this is what we want to worry about. And we actually have a preliminary model. I'm not going to tell you about it, but it's essentially along the lines of what we've seen so far. There is a complicated signaling structure that happens for quorum sensing, interacting cues, chemicals being created, chemicals binding. It's more complicated than what I've shown you. The good news is that it looks very good. The bad news is I'm not sure we can validate it in its current form because I'm not sure there actually is a bacterial strain that has only a single signaling mechanism to induce quorum sensing. In the Pseudomonas bacteria, it's actually 4. And so, which means, so for each signaling mechanism, there's about 3 or 4 cues, right? So if I've got 4 of them and they're interacting and all those other stuff going on, that's when I'm going to really need these sparse approximation strategies in order to solve things. So there's a lot to do. As I mentioned earlier, I think every biological application induces unique constraints. And I contend that maybe Shannon was wrong, that maybe information theory can tell us something about these biological systems because we now are in an environment where we have access to much more data and many more measurements. And this is some of the most fun I've ever had in terms of research, interacting with these incredibly brilliant researchers, but in a very different field. And I would highly encourage it. But having the right collaborator is extremely important because they have to be willing to be nice to you because it might be 2, 3, or 5 years before we can do anything that's actually going to be helpful for them. And I have challenged Mo that when we get this worked out, I would be happy to be the least important author in the list of authors in the Nature or Science paper as long as I'm there. So we'll see what happens. So I have a lot of colleagues that I've worked with. So over here on the left is the USC side. So Nicolo is my amazing postdoc who's going to become an assistant professor at Purdue in January. Sahand, also quite amazing, a physics PhD student who just graduated. He works with Mo El Nagar, who's my initial collaborator here. And James Bodeker here is the new fellow who's working on quorum sensing. And then I was fortunate enough to have Masume Nasiri Kanari come on sabbatical with us at USC. And she had already written a couple of these papers on molecular communication, so in a way she got me involved with that. Of course, Amin Gohari is somebody that I think a lot of the information theory people know. He got his degrees at Berkeley. He's a young assistant professor at Sharif, and a very nice collection of really bright graduate students who work with us. So I'm almost done. I need your patience just for a few minutes because I have 2 pieces of shameless advertising that I need to do. So the first is for the journal. I would really encourage you, if you are doing any work related to biological systems, even though this is a journal out of the Communication Society, I firmly believe that in biological systems, the distinction between communication, sensing, and control is very thin, and that we should be taking a very holistic view in terms of our understanding, designing biological type systems. And so please do submit. And then the last piece of shameless advertising is that I'm looking for postdocs. So I'm also looking for graduate students, but I would not dare steal anybody from KTH. But if you have interests or know anyone with interest in either communication control interface, sparse approximation, or queuing theory, they're not all on the biological stuff, but we have a large number of other projects. And as I said, I'm about to lose my great postdocs, so I need to find at least one, but probably 2 new ones. I really appreciate your patience. Thank you for coming.